Thank you, David. That's a very nice introduction, and thanks for inviting me. Today I will kind of discuss and describe a project that's been going almost now probably 6 or 7 years. This is one of the first collaborations that I developed when I arrived in the UK. As David said, I was in Paris 10 years ago, and then in 2007 I moved to the University of Bristol. So at that time I met Craig, who has a lab in Bristol, and we started looking at gonadotrophin-releasing hormone signaling. I will describe in detail the system and the problem, and this is work that has been ongoing since then. So there are like two main parts to my talk. One, the first part is going to discuss modeling of pulsatile GnRH signaling. I'm going to explain what GnRH means. And the second one is more recent work, which is in its kind of beginning. We've been doing this since last couple of years. It's on quantifying reliability of signaling. Since this project has been motivated by a real biological question, this work has been done hand in hand with experiments. So there is a lot of the content of my talk will be really showing data and explaining the system and how this data is being used in the model, and how the model is being used to generate more experiments and to inspire further work in the lab. So the system that we're looking at is we can call it reproductive system, but this is really just the control of the system that controls reproduction, and it is done by a specific cell type in the pituitary gland, which is shown over there. They told me that not everybody here, most people are mathematicians, so I'm going to go in details over the biology.
Sure.
Yeah.
Go ahead.
So its pituitary gland is over here, and it contains a mixture of cells, different cells which secrete important hormones, and one of them are these so-called gonadotroph cells that secrete this first. I'm going to call them LH and FSH because the actual name are Latin name are very long. These are known as the first hormones of reproduction, and this gland, which is very close here to the brain, is controlled by neurons located in the hypothalamus, called GnRH neurons. GnRH stands for gonadotrophin-releasing hormone. So this is a hormone that's released by the brain to control or to kind of stimulate the cells in the pituitary gland, which then secrete the reproductive hormones. And this process happens in a pulsatile manner. So the system is highly dynamic, and just to connect this to the whole body homeostasis, these hormones LH and FSH then stimulate the ovaries or the testes, depending on the gender, male or female, and this in turn has its, So there are further kind of regulators, hormones that are secreted by these glands, which known as sex steroid hormones, and these then feed back to the brain. So it is a kind of shorter and longer time scale feedback system. However, for the purpose of this talk, I'm going to look at the regulation of secretion of these first hormones of reproduction, the so-called LH and FSH. So the release or the controlling hormone, the GnRH hormone, the gonadotrophin-releasing hormone secreted by the brain, acts on a specific receptor located on the gonadotroph cells called GnRH receptor, and it's a G protein-coupled receptor. This is a class of receptors which are found throughout the body, and the GnRH receptor belongs to this class. So here, this is a simplified representation of the the complexity of the signaling, intercellular signaling that gets triggered or activated upon stimulation of this receptor. Since it belongs to the group of G protein coupled receptors, there are some classical pathways, intercellular pathways, and this kind of diagram tries to capture the most commonly shown to be activated and triggered upon activation of G protein coupled, in particular GQ protein coupled receptors. So we have stimulation of calcium release from intercellular stores via IP3 dependent pathway, stimulation of PKC. These early kind of events act on secretion, which is up there, of reproductive hormones, but they also have downstream targets which activate transcriptional factors such as NFAT or ERK, which stands for extracellular regulated kinase. In its phosphorylated form, it is known that it translocates to the nucleus and then activates expression of transcription of proteins that act as transcription factors. So it's a very complex intercellular signaling pathway, and yeah, this is just here to say that there are over 400 target genes which trigger or participate in the expression and subsequently production of these hormones. Alpha GSU stands for gonadotropin secreting subunit, and LH is the beta subunit. A is alpha subunit of this, and FSH beta is the other beta subunit of the FSH. So they share a common alpha subunit, and they have distinct beta subunit, the genes.
So Khasi,
yes?
This is inside the neurons?
No, this is inside the gonadotropes.
Oh,
One
okay.
in the pituitary cells.
Which are where?
Which are close to the brain, in the pituitary gland.
So they are
Up
different.
there.
So they are different cells.
They are here,
This
and
is
they
the
are
blue.
over there.
In this scheme, this is the blue ones.
No, this is like a little bag full of cells,
Okay.
and they are not only these ones. Here you have the ones that secrete growth hormone, the ones that secrete the ACTH that's related to the stress response, and you have also the ones that secrete the thyroid gland.
Okay.
hormone.
So they are stimulated not by neurons.
They are these projections. See, these are the neurons, the generating neurons that project to the just here. I think this is the brain blood, the barrier that's,
Okay.
that separates the brain. So these project through it and stimulate.
So the neuron, through the liberation of neurotransmitter.
Generate, yes. That's like a neuropeptide.
Okay, that's activate directly those cells.
Yeah, via the G protein coupled receptor signaling
And then when they
over here.
and when they secrete, this is a vesicular type secretion.
Yes, it is.
And then where it goes, the molecule that
This
activates-
one,
Yes.
this goes into the blood, and then through the blood goes to
Okay.
your
Right.
gonads.
Okay.
Yeah, they are. Yeah, they all go in the blood. The stress hormones, the growth hormones, there. Okay, so this system is highly dynamic. That is the point, and the function of the system depends on dynamics. That's the point I was trying to make here with this slide. And this being long known, this is the classical kind of system that's being used to illustrate the importance of dynamics for signaling. Basically, what this shows, and this is a experiment from 70s in monkeys, that if you stimulate the monkey with a pulsatile signal, you get more secretion, and if you expose them to continuous or constant stimulus, the secretion of these hormones is suppressed. And this property of the system, I have another slide, but this property of the system, yeah, it's been usually represented as a non-monotonic, expressed as a, like conceptually, as a non-monotonic frequency-response relationship. Here we have the frequency and the response or biologists call it bell-shaped frequency-response relationship. And it's known that during normal physiological function, this is what stays there. You need increase in the response for ovulation and for proper functioning of the system, and then suppression is used in cancer therapy of treating steroid-dependent cancers in the pituitary gland. So they are medically exploiting these properties of the system already. Also, the IVF therapy is also relying on these kind of properties. And I was really surprised to find out that although clinicians do this in clinic, they don't really know how this works in the cell. There is no kind of, and I'm not going to claim that we have completely answered the problem, but that was the motivation. The question, how does it work on cellular level? But our work has been motivated by the kind of the goal or the aim to try to understand better how this works on intercellular level, and how this signaling pathway, how this kind of dynamic messages or dynamic inputs are kind of interpreted by the cells to produce this kind of outputs, which are also dynamic and are very important.
Excuse me?
Yes?
The full range of frequency is observed in normal conditions, or is it only for therapy that is really used?
The frequency of the signaling from the neuronal level, the hypothalamic neurons, does vary because you have the cycle, but again between species it's very different. And for clinical purposes, they usually go to the extremes because they know that if they drive the system hard, it will have an effect. That's why for the IVF they deliver generation pulses with the kind of optimal, the highest, the one that's known to produce the highest response. In cancer, they would just give a huge dose of constant generate to suppress. So the fine details and the modulation of the system is the one that's kind of still unknown in great detail. So I did talk about this, the optimal stimulation, but what was kind of new, and I discussed this, newly discovered that at the time when I started looking into this about 6-7 years ago, was that this sort of non-monotonic relationship between the response and the stimulation is also could be observed already on gene expression level. These are these subunits of the genes for the two hormones that I was describing, and in my collaborators' lab in Bristol, they have kind of been able to measure, just deliver pulses with different frequencies, and they measure the level of the expression of the genes. And these here are examples showing that even on gene expression level, you can find this sort of non-monotonic and optimal kind of response. So that was another kind of reason to try to go and understand how this may be happening. That's this first part of my talk, where I'm going to describe a model that we developed to try to understand the non-monotonic frequency response relationship of gene expression. And the data that they were generating in the lab was related to measuring activation of two of these many intercellular signaling pathways that are activated, major signaling pathways. One of them is this extracellular regulated kinase. So I mentioned that upon phosphorylation it translocates into the nucleus, and they have a kind of high content imaging system, and they can measure many cells, and they can quantify how much of this kinase is activated. So that is one of the readouts that we are using, and the other one is the activation of NFAT, which is another transcription factor, and is activated by calcium via calcineurin. And similarly, when it's activated form is dephosphorylated, but it's a detail, biological detail. When it's activated, again it translocates into the nucleus, and this translocation could be measured using the same system, lifestyle imaging system, and the amount of activated NFAT and phosphorylated ERK could be then quantified from individual as well as populations of cells. So to just outline briefly, describe the model, based on the availability of data, we decided to model these main pathways, and this is kind of schematic representation of the things that are in the model. So we have activation, we have the hormonal signal that arrives in pulses, and then we have the binding of the hormone to the receptor and activation of GQ. The G protein coupled receptors activate GQ proteins and then activating a receptor. That's the effector, sorry. That is this equation. It's a kind of generic effector that would then activate downstream molecules. One of them, so we have calcium release from intracellular stores, and the effector here is E activating one of the major readouts, the phosphorylated, activating the extracellular regulated kinase, or so-called ERK, via this dual phosphorylation process. And we have calcium, on the other hand, activating the NFAT. So basically, the outputs of this model could be compared to the data that they were measuring in the lab in terms of the activated ERK and in terms of activated NFAT translocated into the nucleus.
But you have a lot of parameters here. Are they
Yes.
known?
Yes. Okay. If it's probably that's a very good point. So the way this is done, this first bit was a model that's, but it's actually very simple one. It's kind of, compared to these ones, which contain a lot of nonlinear functions. This is nonlinear, but not that much. Basically, this model, it wasn't made from scratch. It's based on previous work done in the field. In particular, the activation of the receptor initially, before this data about the non-monotonic frequency relationship on gene transcription level, everybody was explaining this property of the system by desensitization of the receptor. Because if the receptor desensitizes after being exposed to stimulation, to kind of stimulation, then you will get non-monotonic. But then they discovered in late 90s that this receptor doesn't desensitize. So this is not the mechanism. And but there were some models that people have already built, which were available. So this is all based on previous model. This activation event. So this is a model that I've worked during my PhD a lot on, release of calcium from the intracellular stores. So I know that very well. This one was a model of NFAT activation in the heart, because in the heart is also important as a transcription factor from good group, bioengineering in Auckland. So I took, that was their parameters. And this ERK model is again from Kodenko's group, which do ERK modeling for many, many years in the States and now I think in Ireland. So again, the parameters are taken from their paper.
So basically, you took all of the parameters that existed.
As much as possible. Okay. Yes.
So
and then the only ones that I had to modify were my translocations, because I will have fractions of these in the cytoplasm and the nucleus, and the translocations were related to the date. So then I had some data, but only a few parameters to kind of account for. So that's how. Otherwise, this model has I don't know how many, but many, 60 maybe more.
Sorry, regarding the desensitization, so you said this receptor is not desensitized?
It doesn't. It is, so some forms of this, in some animals, they do. In humans, it doesn't. And in mammals, I think in general, it doesn't. But it came later. First, in the 70s, they showed this property, and then there were models from the 80s explaining it using, predicting that desensitization of the receptor will explain. And then they did more experiments, and they showed it doesn't desensitize. So this gets scrapped.
Do you know it's a specific case of the GNRH receptor? Because most
yes,
of the
I know.
are
They do, yeah. That is a very special thing. And actually, in one of our latest papers, I'm not going to discuss that, but they in the lab, they are doing this with different receptors where we quantify the information transfer through the signaling pathway. And yeah, they're looking there with receptors that sensitize and don't desensitize. But yeah, that's true. It's a very special property for this one.
So what is the ratio there? It's non-linear function, c squared or constant plus c squared, which you have in the
This
calcium
one?
equation. Yes.
This is the pump, the plasma, well, this is the circle, that's the ATP pump on the intracellular stores, which pumps calcium back into the store, and that is the flux through the plasma membrane pumps. They are ITP dependent and they need to bind calcium. The second power comes from the fact that it's a cooperativity. They are working in a cooperative manner. But that's a phenomenon, having said that, this is widely accepted way to represent these kind of fluxes. However, it's a bit phenomenological because the actual system is much more complicated. Like, for example, ATP here is not taken into account at all. It needs ATP. And yeah, it's just, yeah. So the data that I used, that really connects to your question, David. We had this kind of data where a single pulse of 5-minute duration is applied to the system, and then this is or constant, and this is the calcium response. This is the pulse response, where calcium goes down, and this is the constant stimulation. So calcium transiently increases, and in a constant level of stimulus, it just levels off like that. And if the stimulus is removed, that's where it's given, it goes down. And the other piece of data, here this one showing the one of these transcription factors called NFAT and its dynamics during, again, a pulse, only short pulse, or constant. In the case of short pulse, it goes up and then goes down. In the case of constant stimulation, it goes up and kind of stays up. So these kind of, I use this as a beginning, starting point to train the model in some sense. And here is just, it's a bit busy, but here are the pulses. This is just a representation of the pulse and the constant stimulation, the first equation, and this here is the response of the NFAT during pulse and during the constant stimulation. And so these are not really fits, they are more qualitative kind of match of this data, and that's the calcium. I'm going to show this again. So more interesting and more kind of important data here is the one that relates to pulsatile stimulation. And here in these experiments, they are giving pulses here of GnRH every hour. That's a realistic physiological frequency. And the duration of the pulse of 5 minutes is also kind of within the physiological range. And they're measuring the activation of the extracellular regulated kinase and the activation of NFAT, and for different doses of the stimulus. And they show quite different dynamics. This is much faster, increasing and decreasing response, and this is slower. I guess that's, and as you increase the dose, you get larger response. In the case of NFAT, and in the case of ERK, it saturates much faster. The response for 10 to minus 9, 10 to minus 7 is almost the same. 10 to minus 10 is slower, is smaller. And these doses of GnRH are also physiologically kind of relevant. Although these experiments are done in cells, in a cell culture preparation. And this is here the model, just qualitatively again, increasing with the increase in dose and kind of showing this time course kind of match as well. And the final one is the frequency response, where now here the pulses are delivered in different frequencies, 5-minute pulses, every 30 minutes to this one, every hour, and every 2 hours. And this is the NFAT, and that is the ERK, again, the time courses. But we can see that this slower response as expected, when the frequency is fast, it doesn't have time to come down to basal level and produce this standard.
Same size pulses here?
Sorry?
Same size pulses?
The pulses are the same size in this case, yes. These are ones are 10 to minus 9. So the same dose but different frequency. And the other one was different doses but same frequency.
So
But when you say same dose, does it mean that same dose over 60 minutes? So if you have 2 pulses...
The
on
same,
one-
the actual pulse is only 5 minutes. You give it,
But
you
it's
step
a-
it, it's a step function, and then you go down, nothing, and then again after hour you step again back to that dose. Keep it for 5 minutes and then go down. And you can do these different frequencies.
Yes, but when you increase your frequency, then you increase the total amount of...
Yes, you do. These are things that we are looking at the moment. Yeah, so you get, I'm not going to talk today, but yeah, because you have a different, because the frequency and the magnitude, they compensate each other. Yes, yes. And there are, interesting things that happen. Absolutely. That's very good observation.
So just to clarify, so those are the experiments where you inject.
Very laborious.
Yeah, but so what you do? You put your stuff, whatever,
Yes,
on the cell, and then you wash.
yes. And
Okay.
then you put again, and then you wash.
And this is to activate. So this activate this G-coupled
Yes, receptor
pathway.
and then these
Okay.
effectors.
And then each time you showed us the very nice.
You do the images, you take the ratio between the nuclear and the cytosol, and then but the machine does it actually, it's a high content.
Yeah, Krasi, my question was that now that you are going to show us, I think in a second, the result of your
Yes.
simulation, and you did it before,
Yes.
you had to adjust some parameter of the model to get from this, or this is just from the...
Here I'm only changing what they're changing in the experiment.
Okay, so all the parameters
So
are
everything is
fixed, are
kept the
fixed
same.
from the... Okay, I
Yes,
see.
yes.
Okay.
And once I should have said it. So once I did this, so here I use this to fix the parameters.
Okay,
The parameters that reproduce this, and then I take this set and only change either the dose, like here, or how much I give, or I change the frequency here. Yeah, that's how I did it. Yes. And then we did all this, and we were really hoping that we're going to be able to explain by having a model and then looking at these activation of genes, how this non-monotonic frequency response relationship can work, but with kind of timescales and responses like that, all you can get is increase as you increase frequency, and you can see you get increase in the response. That's what happens. And this is experimental data of the actual NFAT responsive element. That's the one on the gene that's activated by NFAT, and it shows that it does indeed increase. And this is a transcription factor downstream of ERK. So one is downstream of this, and the other is downstream of that. They both do this so they kind of agree with this, but they don't really explain how this happens, how after or downstream of these ones, one can get a non-monotonic versus monotonic response. So to kind of address this in the models, because that was the limitation of the experimental. So from now on, whatever I'm showing in the model is just from the model. This is the limitation of the current experimental kind of possibilities that can be done in experiment in this particular system. So we kind of hoped that maybe we'll find some negative feedback that would explain downstream of the receptor activation. This we didn't. So we went further to model the activation of these transcription factors, which adds a few more equations. Okay. And we did this in two kind of scenarios. In one, there is a effector like the ERK that activates one transcription factor, and then this transcription factor activates another one, and then they kind of activate expression of genes, or they act on the gene to get expressed. And then there is another one, we call this sequential, where two in parallel, as we know happens in the system, activate in parallel two transcription factors, and then those two somehow act on gene expression. I say somehow because we looked at different ways this could happen as well. So first, the activation of this transcription factor is just Michaelis-Menten kinetic. there are lots of ways to model this. We didn't, yes, that's kind of probably the simplest possible way that one can, other than linear, as it's been done in some papers, that one can, yeah, and it's questionable how appropriate for, in many cases, this kind of framework is. It's what we have done. So we have an activation of these transcription factors by the effectors and the subsequent transcription factors using Michaelis-Menten kinetics minus some degradation of those with the degradation constants. This adds a few more parameters that these are unknown now. So, and then combining those, we used, in the literature it's proposed that the genes can act, the different, yeah, there are conceptual kind of models where two signals can combine in a kind of OR logical gate, so their activity can sum up to promote further events. In this case is the expression of the gonadotropin subunit genes, or they can work in a multiplicative manner, so we looked at summing them, multiplying those activations, and another one where they interact in a more nonlinear, complicated fashion, and that's the function that one of the students that I worked with derived, where we have assumption that these two transcription factors first have to interact before they can activate the gene. So we had these three models, candidate models, and these are simulations of the outputs. Here, the different colors correspond to different frequencies, 4 being the 15 minutes period, so every 15 minutes delivering a pulse, and here the frequency is decreasing, and these are the different, the sequential and the parallel pathways, and the different ways these signals combine to activate the expression. And to kind of sum up, these two, as you increase the frequency, they show increase, as the red gets higher of this activation. This cooperative gate, as we call it, for some parameter values, does show this non-monotonic effect, because you get less activation for the highest frequency. And that happens in both models. And then there is like an optimal sort of frequency, which is about between 1 and 2 hours, where you get the maximum effect, which corresponds to kind of the what's known from experiments. And then we quantify this. These are just quantifications of these graphs. If you quantify this using the area under the curve of these simulations, you get the monotonic increases and the non-monotonic increases, respectively, for the different models. And here, this is just to show that this doesn't come from the transcription factor activations. It's a genuine effect of the way they interact. Yeah, and then this here is just a parameter sensitivity, or just looking at how the parameters that we don't know affect the behavior or the output of the system. And we have these sensitivities of the gene expression for the two transcription factors given by these KD constants, which are here plotted on the X and Y axis. And also the system is sensitive to the degradation type or the decay in the equations. So, and here on this vertical axis, what I plot is the optimal frequency, which is, yeah, this is high frequency and lower frequency, and the various colors correspond to the different decay times. And then we have also, you can see that there are combinations of these parameters where we get optimal response at lower pulse frequencies of stimulation, and there are other sets of parameter values where the system behaves, the responses. I know it's a quite busy graph, behaves monotonically. But basically, this just shows that the system is the output depends on the non-monotonic frequency response relationship requires differential sensitivity of the gene expression to the two stimulus, the activations, the two transcription factors. So we need to have a low sensitivity to the first, here this is small numbers, and higher to the second one in this particular case. Same is for this, and this is related to the time scales at which they evolve. Basically, if you are less sensitive to this and high sensitive to that one, then you obtain the non-monotonic frequency response relationship. And this is kind of concludes the first part of my talk. So we kind of show that the non-monotonic frequency response relationship is kind of could be predicted using this more complicated convergence of two, in this case, two activating factors to stimulate gene expression. And this result with the differential sensitivity suggests some sort of plasticity and possibility to modify the system. The kind of fact limitation, these models are all only valid for kind of population-based measures, and in reality, real systems are characterized by high cell-to-cell variability, and also there is noise. So that's these questions have been motivating those two here, the kind of the directions that we've been taking more recently, how to measure the information transfer and kind of account for the variability in the system. And now this is I can kind of going to switch here from showing you deterministic models to kind of more statistical approaches. Hopefully it's not going to be very confusing. So this approach of applying information theory to cell signaling has been becoming popular in the past few years. And it is really thinking about cells as information channels and asking questions as here, how much we can actually infer about the signal giving the output. Usually when you do experiments, you measure something about your system. And this is in various contexts. This is interpreted as sensitive, like the environmental effect on the system, or how sensitive the system is to the environment, or in our case, we're thinking about sensitivity to stimulation by hormones. And we're using this mutual information measure, which is given in terms of entropy. This H stands for entropy, and distributions. Condition one is the distribution of the output, is the Z is the output, and the other is minus the entropy of the output condition on the signal. And this is just given the formula here. And this could be applied to the data directly using this high content imaging data. In the previous modeling, we just used the averages, and in this case, we're using the outputs measured for single cells to calculate this mutual information from the data. So yeah, so basically we are saying you can use these measures to kind of think about the reliability of hormone signaling, and we're doing this in the context of GnRH. And yeah, here there is something about whether information is lost or not when you move down the signaling pathway. It's interesting to hear your opinions about that after we leave the talk. But you may think that due to noise and lots of various binding and unbinding effects that occur, you would be losing information as you go downstream. Yet biological systems use this kind of regulation and do perform functions. So it is interesting how they do this and protect the against kind of losing information. That's what this is trying to say. So I did mention it from experiments. So for each, this is just to illustrate kind of the setup. With this system, you can measure the response in terms of, now I'm going to focus on extracellular regulated kinase. That's one of our first signaling pathways that is activated by GnRH. And you can do this in all sorts of different situations. It's only with constant stimulation. The pulsatile stimulation is more exciting, but it requires, it's very labor intensive, and they're currently trying to develop optogenetic system so that they can deliver these pulses automatically, but it's not yet working. This is all done in constant, so but you can do constant for different durations of time, 5, 10, 20, and you can then measure, increase the dose, and you can measure the response in terms of dose-response curves. Are these this is clear? Here, we increase the dose of the stimulus, and we measure the output, and this is for different durations of exposure. And then
That's
you can
for average over many cells, or is there single cells?
single cell. Then you can make the distributions from these measures, and then you can calculate these quantities from the data. That's what's, yeah. And then here, this is just the latest one example of the type of analysis that we've been doing. This is, as I mentioned, activated ERK, and then in different conditions with different, these are 2 different signals. Basically, this compares the effect or how much information is passed through the system given to different stimulus. That's a kind of physiological one, and that's artificial chemical one, like a drug. And this is here the effect on gene expression level. This measure here measures the activation of the gene, this fluorescence measure. And we can see that there is a difference between the dose-response curves, and there is a big difference in terms of how much, which happens much slower. As we can see, after 360 minutes you can detect this because it's on gene level. However, you can also see the big difference, and that this is reflected in the mutual information that is calculated from these kind of experiments. In the case of this artificial drug, the system is activated the mutual information is higher, and the response and gene expression level is also much higher. That's just showing some relevance.
What
And,
kind of stimuli is it in this case?
Sorry?
What kind of stimuli is it in this case? Is it continuous?
GnRH, one of them is GnRH.
Is it continuous? Or
Yeah, it's constant. Yeah, it's all of these are done constant, but for different times, 5, 15, 30, and so on. So you can. That's why you can only measure this kind of response on gene level after 360. After 5 minutes, there is nothing. After 15 minutes, there is nothing. It needs some time to propagate down to.
But I'm confused because it ERK decrease through time. As first plot show.
Yes, it does. You get the maximum response very quickly. It's an acute one. However, yeah, as it goes downstream, yeah, it has a delay. Exactly. And the main difference is that you get with this drug artificial one, you get much more. You kind of drive the system much more. So you get more activation, you get higher mutual information, you get higher gene expression as well. And this could be done in models as well. I mean, this kind of calculation of the mutual information, and the analysis is based on work by collaborators of mine. This is just a much simpler model, dephosphorylation cycle of in this case we're thinking of work, molecule protein, and it uses stochastic kind of differential equations and Gillespie algorithm to perform the simulations. And in this paper, where the whole idea is described, the message that came out of this paper was that having the mutual information or the transfer of information could be reduced or modulated in 2 different ways by negative feedback. So here we can see that increasing the feedback strength, the negative feedback strength in this particular model can it initially increases and then decreases the mutual information, but it's also very sensitive to the basal activity in the system, basically increasing the vasoactivity decreases the mutual information as well. And we, in order to kind of look at our particular system with feedback mechanisms on the intracellular and nuclear level, we modified the model a little bit further, including this fast feedback is the one that comes just straight after activation of this effector in the cytosol. And the slow one is due to expression of genes in the nucleus, which then feed back and suppress the activity of this particular protein. So we consider fast and slow feedback, and yeah, these are the kind of the equations that are used. And then we look at how the fast and the slow feedback affects this quantity. This is in the model, and it shows this kind of, again, non-monotonic, in this case, non-monotonic dependence for depending on quite a lot of things, for how long, for how much of signal you have, and how long the system is being exposed to that signal, and also the magnitude of the feedbacks, the slow and the fast. So this is a combination of having fast feedback for here on the x-axis in the presence of slow feedback, and then different durations with different concentrations in color. Basically, the message here is that the information transfer could be affected by these feedback mechanisms in a very kind of non-linear and interesting way. So, because I don't have much time, when do I have to finish? Exactly at 4, or it's 10 to?
Go ahead.
Yeah. Okay.
Don't worry.
Well, I kind of state this, that we can get, depending on the combination of strengths of these feedback mechanisms, we can reduce or increase the mutual information due to various kind of, in terms of interpretation. The negative feedback could reduce the dynamic range, and that decreases the mutual information, but also negative feedback can protect mutual information by reducing cell-to-cell variability. Basically, in the presence of negative feedback, cells are behaving more uniformly, so that leads to increase in the mutual information. But on the other hand, the dynamic range could be. And this here shows a few experiments where we looked at, in the lab, they did manipulations of these fast and slow feedbacks, and kind of estimated or measured in the system the mutual information. So in this first one, the fast negative feedback is being manipulated by this catalytically inactive ARC, which they can make in the lab and put in the cells. And then in this case, reducing this negative feedback basically reduces, as we can see here, reduces the mutual information. This is just measured for different times. Then this here is an example of the effect of manipulating the slow feedback, the slow negative feedback, and in this case, increasing this feedback. These are just the measurements done. These are phosphatases that are expressed in the nucleus and then dephosphorylate ARC, so inactivated, in other words. And this is showing the responses in the presence and in the absence of this kind of manipulation. And I think here is the quantification. So basically here increasing the expression of these dual phosphorylated, dual expressed phosphatases, again impairs, or in other words, decreases this mutual information. And finally is the final experiment with manipulating the synthesis of proteins, which is again using this CHX drug, which affects these feedbacks, because neosynthesis of proteins is required for expression of these phosphatases, which then activate or regulate the slow feedback. And in this case, the preventing the protein synthesis, and in other words, stopping or reducing the slow feedback can improve the mutual information. That is shown actually here. So these are the ones. But it only happens after a long period of time because it involves synthesis of protein. In the beginning, it's the opposite. And this is a summary of these effects that I just described. Basically, decreasing negative feedback can decrease mutual information, increasing it can decrease it, and decreasing it does experimentally causes increase. And conceptually, one can think of the system as sitting on a non-monotonic kind of curve, which describes how the mutual information depends on feedback strength. And these are the different manipulations, which in the case of these feedbacks leads to decreasing mutual information, and this last one leads to increase. To kind of put this into a more, we use modeling now, in the last slide I'll show you, in a more kind of solid ground, we use the model that I described to simulate all these different experimental conditions, and this is here plotted the fast and the slow feedbacks, increasing the fast and here decreasing the slow, and these 1, 2, 3, and 4 are here shown on the graphs. What we see is that in this plane we have a region where we can get an optimal mutual information or transfer information, and the dependence is again non-monotonic. In terms of population average, however, if you just look at the population response, this is not the case. It's a difference in some sense, which is to be expected between population average measures and measures that are obtained from single cells. And this is just a summary. I mean, most of the things that I said, what I showed you is that we use combinations of experimental and mathematical modeling to look at mechanisms that regulate frequency decoding, and we found or proposed that it could be could be like an emergent feature of the networks involved, that the system could be plastic, and finally we are starting to look at information transfer in the system and how it could be modulated or increased in experiments using models and data. And these are the acknowledgements. I showed you a lot of experiments and a lot of data collected over quite a few years. So these are students and postdocs in my collaborators' lab in Bristol, Craig, and this was a postdoc in Craig's lab, and then he's now academic faculty at University of Bath. And these are the modeling and analysis students and postdocs that we've been working with over the years. Thank you very much.
So questions?
Yes?
Concerning the first part of your talk, do you think there is any hope to validate the kind of study predict that
Yes, yes.
there must be some cooperation between the genes?
Yes.
The transcription factor. So if you block one or
That's
mutate
what-
one or express one.
We're at the end of this grant. I've put this acknowledgement to that cancer, research council. We got a grant to actually exactly do this. It's been hard, but some progress is being made because you need to actually do this manipulation on transcription, on gene transcription level. And the main also thing is to be able to stimulate automatically, because this thing with pipetting and washing is really, not to mention that you get also mechanical stimulation from the wash as well when you do this to the cells. So yeah, we're working towards this, and we need more money, I guess. If they give us, we are hoping to. That's the whole point.
So actually I have a question that has nothing to do with the talk.
That's okay.
No, don't worry. At the beginning,
Yeah.
you show the neuron's activity,
Mhm.
which has some kind of oscillatory pattern. I guess this is to stimulate
Yes.
those
These
cells.
ones, yes,
What do we know about the genesis
of this.
of this oscillatory pattern, and do they feel the feedback of the hormones?
Yes, it's all a very good point, and people are working on this actually. So there is some models that have been done, and there is some experimental work. In the hypothalamus, it's a very good question because these neurons are not next to each other. They're far away from each other. They have these axons that somehow produce this absolutely synchronized and precise stimulation to these other cells that I talked about. So there's been models and suggestions that there is some sort of extracellular kind of diffusion of messengers that synchronize them. There is also there is a feedback that comes from these sex steroid hormones that go back into the brain, into the cells. One of them is estrogen, for example. It has a receptor, it's a nuclear receptor that activates genes directly, and it's expressed in many tissues, including the brain. That's one of the mechanisms. It's another very good problem for people. And there are some groups working, but it's not that many, but yeah. So it is, it's an open question, yeah.
The second half showed the feedback effects on information transmission. Is there evidence of these feedbacks, or is it more sort of hypothesized that it could have these effects?
The feedbacks are there, we know. So maybe it was a bit confusing because in the beginning I said there are no feedbacks. It's not that there aren't. It's just that the feedback that are there are on the population level at least, and the way what the measures were showing, they weren't. One of the reasons could be the fact that in the dynamic situation of stimulation, these feedbacks behave very differently compared to, and it would be, because they don't have that much time to affect the system the way they would during constant stimulation. But this activation of these ERK and various others, it goes through, it's a cycle, it's a phosphorylation, dephosphorylation. The dephosphorylation is also already negative feedback on that module. Then you have these other downstream molecules that get activated, and then some of them are phosphatases, so they go back and they dephosphorylate as well. So there are all these feedbacks are in there, and and the question for the dynamic kind of scenario of stimulation is completely open, and I'm very interested. That's what I'm hoping they're gonna fund us, so that we can really ask the question: what happens with these feedbacks, and how important and when are they important? When we have dynamic, which is much more realistic, the system is dynamic. Many of this work of mutual information that has been done in the past few years is in constant stimulation. Right. And very, very recently, a few months ago, there was a paper starting to talk about dynamic mutual information measures from somewhere in the States. But they are starting to
was
look at
that,
those. The system that they do this usually EAST. Yeah. Yes.
I have two questions. The first is, what is the physiologically relevant frequency that you see in these pulsations? So
between 1 and 2 for optimal, hours.
So one signal per 1 hour, one signal per 2 hours. And this is happening in all of us.
It is
Throughout
happening.
day and night.
Yes, it is.
Okay.
There are loads of pulses that happen, not just your heart as well beats.
Yes, but it's just a frequency that you never hear about. So there is a heartbeat which you hear about. There is a circadian rhythm.
That's true.
There's a period in women, but somehow.
But also for male, these things are important too. Although I don't know the details of, but I know that they are important. Well, also the prolactin. Prolactin is the one that activates milk in women, but it's also very important for men, and it happens all the time, and it's also versatile. But I agree, you don't hear about these things. And especially in mathematical communities, people don't really know much about this yet.
Okay, and I have another comment or question to the first part, especially your conclusion. So you had, as far as I remember, 3 different candidate models.
Yes.
One with negative feedback. And what was your conclusion there? So did you
No,
find out which is the best?
best?
The
or-
one that does where the more non-linear one, if I have to give a mathematical answer, is the one where we assume they interact before they kind of act on the system. So there's a non-linear interaction between the two transcription factors. Just one more
Can you
step.
show the
Yeah, I can
slide?
show you this. Yes. I can show you this one. This is the equation which describes this final output that I'm kind of looking at, and this is the function. The other one, I just had the two Michaelis-Menten term sum and multiplied.
But I think there was a slide before this one where that you had some three columns of
In this one?
only
Here?
this one. Yes.
Yeah. So here, you know, what's called integrator could be different things. So I looked at three possibilities for that one. So I looked at two kind of sequential and parallel, and then for each, three.
Oh, so there are three integrators and
And
each
two.
for two, so there's two
Yes,
times two.
yes, yes.
And then, but then what?
And
So
then the
what is the
conclusion
logical
is that
structure?
just the most complicated integrator does it. That's the top one. And the way you can think of this is the red, which is the highest frequency, is lower than, let's say, the purple, which is, this is 30 minutes, that's 1 hour. Sorry. This is 4 pulses per, this is because this is in frequency. So that's pulse per hour. So 4 pulses, 2 pulses, 1 pulse per hour, that's 1 hour, and that is 2 hours. Yeah. So here on top, the highest output you get for those two. And similarly here, this is slightly different, but it depends on the parameters. You can modulate those outputs.
Well, what are you looking at? So what's your objective? Or what
What's
is-
my output is the area under the curve of these traces.
So this is.
Oh, you can
the
take the area under the
GS3.
curve, or you can take just the integral at the last point as well.
So which gives the highest output or
Yes.
highest GS?
The highest, in terms of concept, in terms of number here. See, this is when you quantify. I got this graph is the integral. So for this frequency, the integral in those three different things is that big, is 1. And then it increases as you increase the frequency, and then for one of the models decreases again, and for the others it.
Okay, but what's your conclusion from this? Is your conclusion that this gives the highest GSU output, therefore this is the best, or
No,
for
our
whatever reason,
conclusion
this
is that,
the real model?
yeah, okay. The conclusion is that the system, this kind of interaction of the transcription regulation of gene expression might explain, and we think of this as an emergent property of the system, might explain this kind of non-monotonic frequency response relationship, because in the model we can do that. We haven't shown this experimentally. In
So
other
you're
words,
saying
you
that-
need some sort of interaction on gene expression level to obtain non-monotonic response.
So
That's
your,
the
data,
conclusion.
so your data is a peaked function for the frequency response, and
Yes.
you're trying to find a model which gives you a peaked frequency.
The same qualitative behavior, yeah.
All right.
Okay.
A short one. A short,
Nico,
a short
like
question.
Can you make any predictions upon like the distance they have to be far away from each other in the loop?
You
So if
mean,
you take the persistence length of DNA into account and stuff, can you kind of make any predictions how far they have to be, or like the probability, the rate of having a loop between 1 and 2 transcription factor?
not really, the model is not at that level. It's more, it's deterministic. So all we can, the predictions that we make are here in terms of how they can be related to, probabilities may be related if somebody does a proper translation into a stochastic kind of gene expression model. But these are like sensitivities of these activations to these transcription factors. And that's kind of prediction that you need different, it has to be more sensitive to one signal compared to the other in this particular dynamical situation to get the optimal, which is down here, to get the system to behave as the real system does.
Okay, Krasi, thank you very much.
Thank you.
So next week, there is no seminars, right, Radek? So we'll resume in 2 weeks.